Folks, welcome to a quick little video on the muscle fiber model. So what we have here, what's pictured here, is a muscle fiber. This is a muscle fiber right here. A muscle fiber is another word for a muscle cell. This, of course, is a skeletal muscle fiber. So not cardiac, not smooth, but a skeletal muscle fiber. Remember that it's called a fiber because it is often long and skinny. Often long and skinny. All right, couple things about it. One thing we can see here is this portion here. This portion right here is the plasma membrane of the muscle fiber. The word for the plasma membrane of a muscle fiber is the sarcolemma. Sarco means flesh, lemma means covering, or like meat covering. Now, outside of the sarcolemma, we can see a layer of loose connective tissue. That layer of loose connective tissue is known as the endomysium. It is going to bind one muscle fiber to its neighbors. That way we can create bundles of muscle fibers, just like how we created bundles of axons in nerves and tracts. All right, what else can we see here? Not too much from this view. We really got to turn this guy around. So let's go ahead and do that. If we rotate it a little bit, we are still going to see that sarcolemma right there. And by the way, notice that I can see the outline of some nuclei, labeling the nuclei in purple because I like purple. So nuclei, because remember, a skeletal muscle cell is multinucleate because it was formed by the fusion of embryonic muscle cells. Okay? And what else do we see here? We see a bunch of stuff. I want to point out a portion of the sarcolemma that I'm drawing a purple little circle around. That portion of the sarcolemma right there is known as the motor end plate. The motor end plate. Now, the motor end plate is the part of the muscle fiber that is adjacent to an axon terminal. So there's an axon terminal right there. The axon terminal, of course, is the end of our neuron. And we see some telodendria. Here's a telodendrion right there. There's a telodendrion. So the motor end plate is where we are going to have the muscle cell meet the neuron. I should probably spell that correctly. Telodendrion. Oh, that was supposed to be an E right there. It doesn't matter. All right, so this combination, let's get rid of all that stuff. This combination of motor end plate and axon terminal, that combo creates a synapse, which is known as the neuromuscular junction. Now, the neuron, the axon terminal, is going to release the neurotransmitter acetylcholine. Acetylcholine is then going to bind, so acetylcholine is then going to bind to the nicotinic cholinergic receptors. Nicotinic cholinergic receptors, which are going to be on the motor end plate. And those receptors are actually ion channels. So when acetylcholine binds to them, those ion channels open, sodium rushes in, we get a gradient potential, we get an action potential, which travels along the muscle cell and gets that muscle cell to contract. Okay, what else do we see? Of course, we can see the endomysium over here. We can see the sarcolemma as well. Let's spin this guy around and take another look. Now we got the interior. I can still see a lot of stuff that I met before. I see my endomysium. I see my sarcolemma. I see a nucleus right here. I see my axon terminal there. I see my motor end plate. By the way, notice how ruffled, how up and down the motor end plate is. That gives us more surface area. More surface area means more nicotinic receptors. More nicotinic receptors means we can bind to more acetylcholine. All right. We can see something else here though, and that's what I want to draw your attention to. Look at all of these pillars, these rods, these cylinders, whatever word you want to use. These guys are all myofibrils. Myofibrils are these rods of protein that take up most of the sarcoplasm, most of the interior of a muscle cell. The myofibrils have actin and myosin in them. Remember from the histology lecture, actin and myosin overlap in a repeating pattern, and we can actually see the striations. We can see striations in here. Dark, light, dark, light. Those are striations. Because we got myofibrils, myofibrils have actin and myosin. Myofibrils are going to be what lets this muscle cell contract. All right. Let's look at one more view. Last one, guys. Last view of this muscle fiber. Then we got one more thing to do, actually. What we're looking at here in this transverse view is stuff we already saw. So I can again see my sarcolemma. I can again see a nucleus. And I can see all of these many myofibrils. All right, we're going to pivot. We're going to pivot back to histology again. In this micrograph, I am looking at lots of muscle fibers. I got 1, I got 2, 3, 4, 5. I got basically 6 muscle fibers. Look at their striations. The striations are especially beautiful right here. You can see those stripes. Now, what I'm really interested in here is the new thing. Okay, well, what's new here? What's new is these guys. Let's get an arrow instead. These guys, these dark guys here, these guys are going to be my axon terminals. Those are axon terminals, which means the part of the muscle fiber that they're near, so like the part of the muscle fiber under him, is going to be a motor end plate. Now, notice that I'm also going to see some telodendria. There's a telodendrion, another telodendrion, a third telodendrion. Beautiful telodendria branching off this main axon up here. Now, one last final thing, guys. One last final thing. The combo of the axon terminal and the motor end plate, that combination is our synapse. That is known as our neuromuscular junction. That's where an electrical signal in the neuron creates an electrical signal in the muscle, which then leads to the muscle contracting. And with that, guys, we are done with this little quick video lecture thing. See you next time.